Good day, everyone. My name is Naja Thomas, and this specific slide introduces the presentation of beta-globin protein. It focuses on its role in sickle cell disease and the potential of CRISPR technology to correct genetic mutation. Again, I am the author and the presenter of this specific presentation, and so let's get started. Here is the illustration of beta globin. As we can see up top, you can see that there is a healthy component, and below is the sickle cell component. So the beta globin gene produces a protein that is essential for healthy red blood cells, and mutations in this gene, it causes sickle cell disease, which CRISPR could potentially correct by editing that gene. Here is some brief history. So Linus Paulings worked in the 1940s and the 1950s, where he linked sickle cell anemia to a mutation in the beta globin gene. It reveals how a single cell amino acid change causes abnormal hemoglobin and sickle shaped red blood cells. So, what is the role of beta globin, right? The role is it's a protein, it's vital for hemoglobin's oxygen transport, and its mutation leads to sickle cell anemia, which is because the abnormal hemoglobin S produces a rigid crescent shaped cells with shortened lifespan. Here are some additional information on sickle cell. Well, sickle cell anemia causes episodes, pain, organ damage, and other complications. And with treatments like blood transfusions and medication, will help manage those symptoms. This is just a beta globin structure. It's an illustration. It shows you that the hemoglobin gene is located on chromosome 11 and that this is actually the beta globin gene's activity. So the beta globin structure, we could look to Max Perutz and John both use the X-ray crystallography to determine the hemoglobin's 3D structure. It includes beta globin, which earned them the Nobel Prizes and advanced the understanding of protein folding and function. So let's discuss the gene for protein. So the beta globin gene, it's located on chromosome 11. So this is the gene for protein continued. And so we can see that the beta globin gene's precise location is on chromosome 11, and its role in hemoglobin production highlights its importance in genetic research and therapeutic developments. So we can also look deep into it, and we can see that there is a portion of it where it talks about or it describes the DNA sequence and a protein sequence. In the normal blood cells, we see that everything is accurately and correct. But in a mutant or sickled red blood cell, we can see that there is some type of mutant in the DNA sequence that translates over to the protein sequence. That is where the issue lies. So why is the beta globin interesting, right? It has a critical role in oxygen transport, as well as its link to disease makes it a key subject for studying proteins function, disease mechanisms, and the potential treatments like CRISPR technology. So I want to first start by saying that the importance of a research publication. So I have two research publications I will briefly just walk through a little bit. And so the first one is focused on gene editing strategies. And so CRISPR and Cas9 has been used to edit the HBB gene in human cells, which offers the potential to cure sickle cell disease by actually correcting mutation via the HDR, which is homology directed repair. So for the second publication, this is for efficient editing. This CRISPR edited stem cells from sickle cell patients, it reduced the sickling and increased normal hemoglobin, showing that there is promise for clinical use with minimized off-target effects. This slide provides resources like videos, gene databases, and the FDA updates on CRISPR's application for use of sickle cell disease and any other further learning tools that you might need for this specific topic. So this is an example of a use case of CRISPR-Cas9. It is being tested to correct the beta-globin mutation in sickle cell disease, with research showing successful gene editing amongst patient-derived stem cells. This is a really, really, really good use case. If you're interested, please provide comments, like, share, subscribe, and get more information on different research topics. Also, this is my references, which include studies on beta-globin gene correction, 3D renders of mutations, and then articles on CRISPR's potential for treating sickle cell disease. And it actually, all of these different references or citations are very supportive in my presentation's claims. Thank you so much. Have a great day. I hope that you enjoy this quick, fast-paced presentation on beta-globin and its application or on CRISPR technology and its improvement in gene editing or finding or that mutation and really understanding the differences among healthy cells versus sickle cells. Hope that you enjoy. Please subscribe if you can, like, share, and add. I would love to hear your thoughts on it. And hopefully I will get an opportunity to talk more about articles, research articles, and how they are useful in upcoming weeks, maybe a couple in like the next month or so, just to talk more so about the research publications that I think are really useful and really good to add value to presentations. Thank you so much. Again, have a great day. Talk to you guys soon. Bye.